Hi, it's Tate, and I'll start off with talking about the structure of the mitochondria. So the mitochondria are located in the cytosol of the cells, and so is glucose. And each glucose molecule has 24 electrons that'll be extracted during cellular respiration. And the first step of cellular respiration is glycolysis, and it happens in the cytosol. And each part of the mitochondria, this is the outer membrane, this is the inner membrane, this is the matrix, and this is the intermembrane space. And in the matrix, what happens here is pyruvate oxidation and then the citric acid / Krebs cycle. And then in the inner membrane, along this right here, what happens is oxidative phosphorylation, which includes the electron transport chain, and chemiosmosis. So the first step of cellular respiration is glycolysis, and this occurs in the cytoplasm of the cell, and it does not require oxygen to carry out its functions. And there are 4 phases of glycolysis, and the first one is the energy investment phase, or phosphorylation. And what happens is 2 ATPs are invested into the process, and it starts with a 6-carbon molecule, which is glucose, and then these ATPs turn into ADP by transferring a phosphate to each of them, and it makes glucose unstable, so then it starts to react. Phase 2 is lysis, and that's where the 6-carbon sugar breaks in half, forming 2 3-carbon molecules. Phase 3 is oxidation, and the 3-carbon sugars each have 2 electrons, which are symbolized as these 2 blue jewels, and then they each have 1 proton, or hydrogen ion. And so 2 phosphates are then added from the cell's environment, and to the 2 3-carbon molecules, and then 2 NAD pluses come in, and they take 2 electrons each, and then that turns them into NADH. And then the NADH, they also take a hydrogen with them too. And then the NADH will carry these electrons on to the last phase of glycolysis. So the last step of glycolysis is ATP formation. And so the 3-carbon molecules, each of them has 2 phosphates. Pretend there's 2 here and 2 here. And each phosphate from each 3-carbon molecule is transferred to 4 different ATP molecules, ADP molecules, making them ATP. And this makes 2 pyruvate molecules and 4 ATP molecules. And then we still have just the NADHs right here. We'll just move these to the side. And because 2 ATP molecules right here were invested in the beginning, the net gain is only 2. So the end products are 2 pyruvate molecules, 2 NADHs, and 2 ATPs. And the 2 ATP are formed by substrate level phosphorylation. So now we're moving on to fermentation, which occurs after glycolysis when there is no oxygen available. So glycolysis already happened. We have our products here. And in the absence of O2, or oxygen, we need to break down more glucose so that it can move on then to pyruvate oxidation, but that's in a later step. So another glucose molecule comes in and breaks into 2, and it has 2 electrons per each one. And then this needs to be partially oxidized, so the electrons need to be taken from these 2 3-carbon molecules to make more ATP. But these NADHs are full because they each have their 2 electrons, so they can't pick up these 2 electrons because they're full. So then these 2 electrons are transferred back to the original pyruvate molecules, and these turn into lactic acid. And then these, once they lose their electrons, the NADHs, I almost knocked over my electrons, they recycle, back into NAD+. And they now can take the electrons from the new 3-carbon molecules, and then they reduce once again back into NADH, and then you obtain a net gain of 2 ATP. And it was important to recycle the NADHs back into NAD+, because we needed more NAD+, to oxidize this new glucose in order to produce ATP. The next step in cellular respiration is pyruvate oxidation. So this will take place in the matrix, in the matrix of the mitochondria. And so one of the pyruvate molecules moves into the matrix through the inner membrane. And keep in mind at the beginning how I said that the original glucose molecule had 24 electrons. So we're going to harvest, and we've harvested four so far. So we're going to harvest 2 more from this pyruvate molecule. And an NAD plus comes in, takes the 2 electrons, and then reduces to NADH. And then in doing so, one of the carbons is, if I could get it off, is released and exhaled as CO2. And then this new molecule is now acetyl-CoA, and it happens again with the other pyruvate molecule. So it moves in through the inner membrane to the matrix, and we'll harvest 2 electrons from that one as well. And an NAD comes in and takes those electrons and is reduced back to NADH. And then this pyruvate also loses a carbon molecule and is exhaled as CO2 as well. So now we have 2 acetyl-CoA molecules, and we have 2 CO2s that are over here, 2 carbon dioxides that are now exhaled, and we have 2 of the NADHs. So we now move on to the citric acid cycle, and this would what usually happens is a 4-carbon molecule called oxaloacetate reacts with the acetyl-CoA and it makes a 6-carbon citric acid. But right now we're only putting emphasis on the carbons that are just from the glucose. So these 2 carbons from one of the acetyl-CoAs are released as carbon dioxide. And then we keep in mind that there are 24 of the electrons in total. So each of the acetyl-CoA, has 8 electrons, and so an NADH or an NAD plus will come in, take 2 electrons, and reduce to NADH, and this happens 3 times. So takes 2 electrons, turns into NADH, and then takes 2 electrons and turns into NADH. So now there are 3 NADHs. And then an FADH, an FAD comes in, which is another electron carrier. That comes in and also takes 2 electrons, and then reduces into FADH2. And keep in mind that we have 2 of these acetyl-CoAs, so this happens once more. 2 of the carbons are released as carbon dioxide, and then don't have enough of the electrons pulled out. 1 second. Alright, I got them. So this acetyl-CoA also has 8 electrons, and so an NAD plus will come in, take 2 electrons and reduce to NADH. Another one comes in, reduces to NADH when it takes 2 electrons. Oh, that's upside down. Oops. If I could pick it up, that'd be great. Go, just like that. And then turns into NADH, takes 2 electrons. And the last NAD plus takes 2 electrons and turns back into, reduces into NADH. And then an FAD comes in again and reduces into an FADH2 and takes 2 electrons. And then each of these 2 acetyl-CoAs with those both gain 1 ATP. The 2 ATPs are formed by substrate level phosphorylation, and the end products are 4 carbon dioxides, 2 FADHs, 2 ATPs, and 6 NADHs. This 3 and 3, and these 2, and then these 2, and then these 4. And then the 2 other NADHs right here, those are from the previous step, and so are these 2 carbon dioxides. Sorry, I just completely blanked on that. Oxygen is required for pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation. The energy intermediates, which are NADH and FADH2, will begin to take the electrons to the electron transport chain. So to start the process of the electron transport chain, oh goodness, sorry, an NADH gets dropped off at protein complex 1, and then its electrons will be right there, And then in doing so, it will pump a hydrogen ion into the intermembrane space because it's energized. And I'm just going to move this NADH down here so it's a little bit out of the way. And then another NADH will come and drop off at protein complex 1 and send its electrons with it as well. Try to get these in a line. There we go. And in doing that, another hydrogen ion will be pumped through to the intermembrane space. And as the electrons move through the protein complex 3, another hydrogen ion will be pumped through. So then NADH comes to protein complex 1, the electrons get added to that chain, hence electron transport chain. My goodness, this is impossible. And then another ion, or hydrogen ion, will be pumped through to the intermembrane space. Forgot to mention, as the hydrogen ions are pumped through, the NADHs are all going to be reduced to NAD+ once again because they're losing their hydrogen ion. I'm trying to grab it all without everything coming off the piece of paper. There we go. Then another NADH will drop off at protein complex 1, drop off its electrons, be oxidized or reduced back into NAD+, and it'll pump another hydrogen ion into the intermembrane space. And then they keep passing through the protein complex 3, so then another hydrogen ion will be pumped, because every time they cross or go over the protein complex 3, another hydrogen ion will be pumped into the intermembrane space. And then as this electron pair from the first NADH that we dropped off goes through protein complex 4, another hydrogen ion will be pumped through, and this chain keeps moving progressively this way to the right because more and more NADHs will be dropped off at protein complex 1, their electrons will be added to the line, and then they'll be oxidized back into NAD+, and then another hydrogen ion is pumped through. So per each NADH, there will be, and as their electrons cross 1, 3, and 4 of the protein complexes, there will be 3 hydrogen ions per NADH. And then for FAD, it's a bit different. This drops off at protein complex 2, the little red one right here. Its electrons join the transport chain ahead of protein complex 1. It oxidizes back. If I could pick it up, my nails are not letting me. There we go. It oxidizes back to FAD, and then as this goes through the chain in each protein complex, it each FAD plus will then transport 2 hydrogen ions. So then, just put these here so you can visualize it a little better. So there'll be 2. As these electrons are moving down the chain in their line, well, we can just pretend that's a line because it's not cooperating at the moment. There we go. As they're moving down the chain, the last electron acceptor is oxygen, and then those get turned into water as they keep going. And then as this process is occurring over here, more NAD will come and drop off its electrons to add to the chain, be oxidized, or NADH, and then it'll be oxidized back to NAD+, and then it will release more hydrogens. And then as it keeps going, more and more hydrogens will be added because these will eventually be dropped off, the NAD+ at protein complex 1 and FADH2 at protein complex 2, and their hydrogen ions will be pumped in as this keeps going kind of in a cycle. And as this whole process is happening, all of the electrons are being picked up by oxygen, and then they're all being reduced into water at the end of the chain. And as that happens, with all of the electrons that were from just one glucose molecule, assuming that it was 100% efficient and worked perfectly, well, not perfectly, but you get what I mean. There will be a total of 34 hydrogen ions in the intermembrane space, and this is the proton gradient. This is a proton gradient or an electrochemical gradient, and so down the electrochemical gradient, this hydrogen ion will be pumped through ATP synthase through facilitated diffusion, and then in doing so, ATP is created from ADP forming with inorganic phosphate. So we will do that again. So hydrogen pumps through ATP synthase through facilitated diffusion, and then ATP is created by from ADP adding with an organic phosphate. Assuming that this worked 100% effectively and efficiently, then since there were 34 hydrogen ions, and as each one passes through ATP synthase, through facilitated diffusion, one ATP is created. There will be 34 ATPs. So 30 were produced from the energy that was harvested and transferred by the NADH, and then, or the NADHs, and then 4 of the ATPs are produced from the harvesting and transferring of energy by FADH2. And an important factor to keep in mind is that we're always losing energy due to heat, which is the law of thermodynamics. And so in reality, we'll be usually having producing 30 to 32 ATP. And these 30 to 32 ATPs are from one glucose molecule, all of this is. And then these are also made by oxidative phosphorylation. It's also important to keep in mind that remember the 2 ATP molecules that were created in the beginning by substrate level phosphorylation. The energy flow begins with the intermediates, and as they go through and drop off their electrons, those go to the energy goes to the electron transport chain, and that'll pump energy through to the hydrogen ions, and then those come around through ATP synthase to ATP. And keep in mind that the electrons are now in a different space. They're now included with water. So when they're dropped off and picked up by oxygen, they are reduced to water.